Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notification. In this video, I am going to explain one of the topic in photosynthesis in higher plants. It is dark reaction related to Calvin cycle. The process of photosynthesis, it occurs with two steps. It is light reaction and dark reaction. In photosynthesis, after completion of light reaction, some more photosynthetic reactions occur in stroma of chloroplast without involvement of light energy. But these photosynthetic reactions occur by utilizing the products of light reaction like ATP and NADPH without involvement of light energy. These reactions are named as dark reaction. The products of light reactions like ATP and NADPH along with oxygen, in this oxygen it diffuses to outside of the chloroplast, but the remaining products like ATP and NADPH, these are utilized in dark reaction that leads to synthesis of food material in the form of glucose or sugars. The products of light reactions like ATP and NADPH, these are essential for assimilation of carbon dioxide into carbohydrates like glucose. During this dark reaction, the energy present in ATP and NADPH is utilized for the conversion of carbon dioxide into glucose. The dark reaction means these dark reactions it not occur in dark, but light not participated in this reaction, so that these dark reactions occur in the presence of either light or dark only. So due to synthesis of carbohydrates from carbon dioxide in dark reaction, these dark reactions are also named as biosynthetic phase. At this time, this process does not directly depends on the presence of light energy, but these reactions are dependent on the products of light reaction like ATP and NADPH. Along with these, carbon dioxide and water also participated in dark reaction for the synthesis of carbohydrates. So after completion of light period, the light become unavailable, so that the biosynthetic process it continue for some time, and then this process it will be stopped. If then the light is made available, due to availability of the light, the synthesis of food material starts again in photosynthesis. Hence, it is also called as biosynthetic phase, as dark reaction is a misnomer. So the dark reaction occur through a special process. It is called Calvin cycle. In this Calvin cycle may be supported by even C4 and crassulation acid metabolism, which take place in certain plants, is also called as CAM cycle. That means the Calvin cycle is the main basis for carbon dioxide fixation, even though in C4 cycle and CAM cycle also C3 cycle takes place. The ATP and NADPH, which are produced in light reaction, these are utilized in biosynthetic phase, and we saw these earlier that the carbon dioxide is combined with water to form CH2O, is taken enzymes or sugars during photosynthesis. This photosynthesis, the process of dark reaction like Calvin cycle, it takes place in stroma. At this time, the light reaction products like ATP and NADPH are utilized for the conversion of carbon dioxide into sugars. The general formula of sugars is CH2O taken n times, and depending upon carbon number, the type of carbohydrates is formed. The scientists, they try to find out how this reaction occur or proceeds, or rather than what was the first product formed when carbon dioxide is taken into reaction or after carbon dioxide fixation. In this, the first method of carbon dioxide fixation is Calvin cycle, is also called as C3 cycle. So just after Second World War, among several efforts by using radioisotope to use the work of Calvin cycle, and by taking these radioisotopes, Calvin he conducted several experiments to know the pathway of carbon dioxide fixation. The use of radioactive C14 by Melvin Calvin in algae photosynthesis, it helps to study and discover the first carbon dioxide fixation product, and it was found out as 3 carbon organic acid. That's why this cyclic process discovered by Melvin Calvin is named as C3 cycle. And he also contributed to work out the complete biosynthetic pathway of carbon fixation, and due to discovery of this pathway by Calvin, it is named as Calvin cycle after him. The first product, it was identified as 3-phosphoglyceric acid. In short form, we can call it is as PGA. And Calvin cycle occur in all photosynthetic plants, either it is a C3 plant or C4 plant or CAM plant also. The primary acceptor of carbon dioxide in Calvin cycle is, the scientists also try to know whether all plants have PGA as a first product of carbon fixation, and the primary carbon dioxide acceptor molecule during C3 cycle, it is with a 5 carbon keto sugar, it is named as RUBP. It is also called as ribulose bisphosphate. So in C3 cycle, ribulose bisphosphate acts as a primary acceptor of carbon dioxide. And the first reaction in Calvin cycle, it was catalyzed by Rubisco enzyme for carbon dioxide fixation. The full form of Rubisco enzyme is ribulose bisphosphate carboxylase oxygenase. Simply it is called Rubisco enzyme. The very most important importance of Rubisco enzyme is, it is the most abundant enzyme present on earth in plants, and it is characterized by the fact that active site can bind to both carbon dioxide and oxygen. Hence, that's why it is named as oxygenase carboxylase enzyme, because Rubisco enzyme have a capability to perform both carboxylation reaction and oxygenation reaction depending upon the availability of carbon dioxide and oxygen. Rubisco has a much greater affinity for carbon dioxide than oxygen, so that it binds in competitive way. At this time, before the scientists discovered the 5 carbon keto sugar, which acts as a primary carbon dioxide acceptor, and they believed that the first product was C3 acid, so that before discovering Rubisco, they thought that the first carbon dioxide acceptor maybe contain 2 carbon molecule, and they believed that the first carbon dioxide acceptor is 2 carbon compound. The stages of Calvin cycle. The Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in cyclic manner, and this cyclic process, it was discovered by Melvin Calvin. That's why it is named as Calvin cycle, and RUBP again regenerated in this cyclic manner to continue the process of C3 cycle or Calvin cycle. The Calvin cycle can be described under three important steps. In this, the first step is carboxylation, second step is reduction, and third step is regeneration. In this, the first reaction of C3 cycle or Calvin cycle is carboxylation. So generally, carboxylation means addition of carbon dioxide. So carboxylation is the fixation of carbon dioxide into a stable organic intermediate compound. In this, the carboxylation is the most important crucial step for Calvin cycle where carbon dioxide is utilized by RUBP to perform carboxylation. So in this, whenever the carbon dioxide is received by ribulose bisphosphate and this reaction is catalyzed by RUBP carboxylase, oxygenase or Rubisco enzyme, and this first reaction it is catalyzed by Rubisco when the carbon dioxide is available. This reaction is catalyzed by Rubisco enzyme and it results in the formation of 2,3-phosphoglyceric acid molecule. Simply we can call as 3-PGA. So 3-PGA is the first product in carbon fixation of Calvin cycle and it is formed with two molecules. So here in this flow chart, whenever the three molecules of RUBP it reacts with the three molecules of carbon dioxide, it leads to the formation of 6 molecules of 3-phosphoglyceric acid. It is considered as first step in Calvin cycle. The second step is reduction. So after completion of carboxylation, the 6 molecules of 3-phosphoglyceric acid is formed and these 6 3-phosphoglyceric acid molecule they undergoes a series of reaction for the formation of glucose or carbohydrates. At this time, the 6 molecules of 3-phosphoglyceric acid it utilizes 6 ATP molecules and 6 NADPH plus H plus molecule which are formed in light reaction and it will be converted into 6 molecules of glyceraldehyde 3-phosphate. And the step involved in utilization of 2 ATP molecules for phosphorylation and 2 NADPH molecule for reduction of carbon dioxide molecule fixed. The fixation of 6 molecules of carbon dioxide it take place in 6 turns of cycles and it utilizes and required for the removal of 1 molecule of glucose in this pathway. These are the steps present in reduction reaction. In this reduction, 6 molecules of 3-phosphoglyceric acid it converts into 6 molecules of glyceraldehyde 3-phosphate by utilizing 6 ATP and 6 NADPH molecules, and after completion of these, out of the 6 molecules of glyceraldehyde 3-phosphate, only 5 molecules are regenerated into RUBP and remaining 1 molecule of glyceraldehyde 3-phosphate it is utilized for the synthesis of carbohydrate or glucose in the form of C6H12O6. And the third step in Calvin cycle is regeneration. So in this regeneration, the carbon dioxide acceptor it is RUBP. It is very crucial if the cycle is to continue uninterrupted. The regeneration steps it require ATP molecule for phosphorylation to form RUBP. At this time, out of 6 molecules of glyceraldehyde 3-phosphate, only 1 molecule of glyceraldehyde 3-phosphate it is utilized for the synthesis of carbohydrates, but the remaining 5 molecules of glyceraldehyde 3-phosphate by utilizing 3 ATP molecule again it regenerated into 3 molecules of RUBP or ribulose bisphosphate, and it is a process called regeneration. And for every carbon dioxide molecule which enters into Calvin cycle, Each carbon dioxide, it utilizes 3 molecules of ATP and 2 molecules of NADPH. After this, it is 2 meet to difference in number of ATP and NADPH that are participated in cyclic phosphorylation. When we count the number of ATP and NADPH molecule in Calvin cycle, so whenever the 6 carbon dioxide molecules enters into Calvin cycle, the 6 molecules of carbon dioxide, it is useful for the formation of 1 molecule of glucose. At the same time, for each carbon dioxide molecule, 3 ATP molecules are utilized. For 6 carbon dioxide molecule, 6 into 3, totally 18 ATP molecules are utilized, and it becomes into 18 ADP molecules. Along with that, for each carbon dioxide molecule, 2 NADPH are utilized. For 6 carbon dioxide molecules, 6 into 2, 12 NADPH molecules are utilized, and it becomes into 12 NADP. In this way, whenever one carbon dioxide molecule, it participated in Calvin cycle that utilizes 3 ATP and 2 NADPH molecules which are formed in light reaction. At the same time, for the formation of 1 glucose molecule, totally 6 carbon dioxide molecules are required, and the process of Calvin cycle occur 6 times by utilizing 18 ATP and 12 NADPH molecule for the synthesis of 1 glucose in dark reaction or Calvin cycle or C3 cycle. Thanks for watching.